Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta from IIT Delhi. Today we are going to talk on module Phospholipids from paper Structure and Function of Biomolecules 2. Apart from carbon, hydrogen, oxygen and nitrogen, phosphorus is an important element in biochemistry. Nucleic acids are formed by nucleotides joined by the phosphodiester bond. As RNA world existed before the DNA world, the role of phosphorus in biological molecules during evolution happened early. Biomembranes contain phospholipids. That also signifies the importance which evolution attached to exploiting phosphorus chemistry. Let us devote this module entirely to this important class of biological molecules which are called phospholipids. So the objective of this module are to learn about the structure and importance of glycerophospholipids, to learn about plasmalogens and their occurrence, to also learn about phospholipases, various phospholipases and their applications in biochemistry. So the concept map consists of all about phospholipids, how are they degraded by phospholipases, what is their importance, and of course what are their structures. And as far as phospholipases are concerned, we will talk about A1, A2, C and D. In glycerophospholipids and some sphingolipids, the polar part of the molecule is attached to rest of the hydrophobic part by phosphodiester linkages. Such molecules are called phospholipids. Not all sphingolipids contain phosphate. Such sphingolipids have sugars instead forming the polar head. These are called glycolipids and will be discussed separately in the next module. Phospholipases hydrolyze phospholipids at specific bonds. The early phospholipases were identified in snake venoms. Phospholipase C and D for quite a while held limited interest. As gradually their wide-ranging roles emerged, their enzymology became quite exciting. Some basic structural information about the glycerophospholipids and other phospholipids has been already provided in the early introductory module. We will build upon those basics after now having acquired little more knowledge about the importance of fatty acid components in shaping properties of the lipids of which they are constituents. The simplest glycerophospholipid are phosphatidic acids. One can view these as derivatives of glycerol 3-phosphate with C1 and C2 OH of the glycerol 3-phosphate esterified with fatty acid acyl chains. They are called acids because of the presence of the phosphate group forming a monoester with the terminal CH2OH of the glycerol. The phosphate group is ionized and can pick up a proton. Phosphatidic acids are present in biomembranes but in very small amounts. When the phosphate group forms a diester with another alcoholic group like that from choline, serine or ethanolamine, the corresponding compounds are named as phosphatidyl choline, phosphatidyl serine and phosphatidyl ethanolamine. Phosphatidyl choline is also known as lecithin. These glycerophospholipids occur commonly in biomembranes. Generally, fatty acids at C1 are saturated C16 or C18 fatty acids. At C2, fatty acids are often unsaturated fatty acids of C16 to C20 chain length. These amphiphiles have the two acyl chains as the nonpolar tails, with one polar head consisting of the phosphate esterified with the alcohol from choline, serine, aminoethyl moiety. The formula of one such glycerophospholipid, 1 stearoyl, 2 oleoyl, 3 phosphatidyl choline is shown here. In the space filling model, please note the bent shape of the oleoyl chain as compared to the straight palmitoyl chain in the molecule. Little later, we will see the significance of this when we discuss the role of lung surfactants in the function of that organ. The nature of the fatty acids present in any kind of phospholipid, like phosphatidylcholine, can vary widely. This variation can occur in the same cell or same tissue or within the same organism. The distribution of the different molecular species also varies from organism to organisms. Diphosphatidyl glycerols also occur. Its most important example is cardiolipin. Two phosphatidyl groups share the same glycerol in such molecules. Cardiolipin was first isolated from the heart tissue, hence so named. It is, however, also present in other tissues of the animals. This phospholipid is a good immunogen. Phosphatidylcholine and phosphatidyl ethanolamine are derived from diacylglycerol. Phosphatidyl ethanolamine can be transformed to phosphatidyl serine in animals in an exchange reaction. The enzyme catalyzing this reaction requires calcium ion situated on the cytosolic side of the endoplasmic reticulum. It is called base exchange enzyme. Some analogues of phosphatidylcholine contains an ether linkage instead of acyl unit at C1 carbon. These glyceryl ether phospholipids are fairly extensively distributed in animal tissues, while in most of the tissues their overall percentage of the even total phospholipid content is not large. Their concentration is much higher in erythrocytes and bone marrow. Interesting enough, in slugs and some mollusks, up to 25% of the phospholipid content can be these ether phospholipids. Their precursor is dihydroxyacetone phosphate, which is the glycolytic intermediate. 2-acetyl ether derivative of phosphatidylcholine is a platelet activating factor. At low concentration of 0.1 nanomolar, it is capable of aggregating blood platelets. It also causes dilation of blood vessels. A simple substitution of long chain fatty acid by the acetyl group in this compound is enough to enhance the compatibility of this molecule to function in predominantly aqueous environment of the blood. This platelet activating factor is released from basophils. As it is discussed in the immunology section, basophils are white blood cells which are stained with basic dyes. Upon being secreted by basophils, it also induces platelets to release serotonin. This interesting phospholipid also affects in diverse ways other organs like liver, smooth muscle, uterus, and lung. It is also involved in inflammation and allergic responses. Plasmalogens are also ether phospholipids. Their structural characteristic is that C1 has an alpha beta unsaturated ether. The alkyl precursor undergoes loss of 2 hydrogen catalyzed by a microsomal enzyme desaturase. The reaction actually is similar to the one which takes place when double bond is introduced into saturated long chain fatty acids during their synthesis. The reaction requires NADH and oxygen and participation of cytochrome B5. Phosphatidyl choline is a plasmogen corresponding to phosphatidyl choline. The ester group at C1 of the latter is replaced by the unsaturated ether. In the name, Y is replaced by A. In vertebrates, nearly 50% of the hard phospholipids are plasmalogens. The other rich sources of ether phospholipids are membranes of halophilic bacteria, ciliated protists, and some invertebrates. Plasmalogens are also synthesized from dihydroxyacetone phosphate as a precursor. The most common bases present in plasmalogens are ethanolamine, choline, and serine, which are added to the structure from their CDP derivatives. In a genetic disorder called Zellweger syndrome, the liver and brain cells are deficient in plasmalogens. The infants born with this syndrome die early. It is believed that plasmalogens play an important role during embryonic development. Edible oils need to be refined before being sold as a premium product. When obtained in their crude form, many oils contain gummy material, and it's their removal which is called degumming. In the classical industrial practice, oil is mixed with water and a degumming agent. Such degumming agents include citric acid, phosphoric acid, oxalic acid, acetic anhydride, or even malic anhydride. A lecithin sludge is obtained which consists of many phospholipids, phosphatidic acid, and some glycolipids, and even oligosaccharides. Depending upon the extent of removal of water content by evaporation during degumming, the lecithin sludge so obtained can be either a highly viscous material or a powder. Industrially, soybean has been the main source for obtaining lecithin. Some other sources like rapeseed, sunflower, and corn have also been tried. Soybean remains the main source because of its functional superiority. Neutral and polar lipids are extracted from the crude lecithin by acetone extraction. Use of ethanol or ethanol water co-solvent mixtures extracts phosphatidyl choline, and phosphatidic acid and phosphatidyl inositol are mostly removed. Lecithin occurs as a byproduct in oleochemical industries, and itself can be used as an oil and water emulsifier in food processing. It is used for margarine production. The higher ratio of the phosphatidyl choline to phosphatidyl ethanolamine makes the preparation a better emulsifier. The insoluble part contains acidic phospholipids and is used in water and oil systems. Viscosity of the chocolate mass is increased by adding this preparation and partly replaces the requirement of cocoa butter. So industry producing chocolate has a requirement for this product, which in itself is a byproduct of lecithin purification process. Purified lecithin is a highly priced product and is obtained by adsorption chromatography with media like aluminium oxide, silica gel or DAE cellulose. Industries which use lecithin include pharma, cosmetics and food. The main utility in all these applications is in stabilizing water and oil emulsions by acting as an emulsifier. The level of purity required varies with the application. The composition, especially presence of other phospholipids and fatty acid content, dictates not only emulsifying quality but also whether oil water or water oil emulsion is to be stabilized. Many cosmetics contain 0.5 to 1% lecithin, which imparts skin feel to these products. Ease of spreading, wetting ability and reduction of oily or greasy feeling are some of the useful results of incorporating lecithin in such products. Industrial coatings, paints, ink contain lecithin as a dispersing agent for the pigment. Bakery, beverage and confectionery products use lecithin as a food grade emulsifier. In baking, it is used as a dough conditioner and aids mixing of fat, flour and sugar. For many other applications, lecithin is modified chemically or by using enzymes like phospholipases. Hydroxylated lecithin, for example, is a better oil and water emulsifier. Regulatory authorities prohibit use of most of the modified lecithin products in processed food. Lung surfactant is a mixture of protein and lipid of which 80 to 90% on a weight basis is phospholipid. About 70 to 80% of these phospholipids is mostly DPPC alone. DPPC stands for Dipalmitoyl Phosphatidyl choline. What makes DPPC a useful constituent of lung surfactants? The saturated long chain of palmitoyl moieties extend without bends. This allows the molecule to pack as a single layer. This orientation in this is that the polar phosphatidyl choline head faces alveolar cells. These are the cells in the lung which form air spaces called alveoli. The surfactant coats the surfaces of these cells. The non-polar layer of the palmitoyl chains face the air. During the breath expulsion, the alveolar volume and the surface decrease. The layer hinders complete collapse of the alveolar space. This also facilitates the expansion-contraction cycle of the alveolar cells. Expansion of a totally collapsed alveoli would be more inefficient in terms of effort required. Alveolar cells continuously synthesize and release the surfactant. Premature infants have very little production going on, hence these babies are more prone to alveolar collapse. Such a situation leads to respiratory distress syndrome. In adults, an injury of the lungs can lead to insufficient surfactant production. The result is adult respiratory distress syndrome. In both cases, giving exogenous surfactant helps. The respiratory distress syndrome, as the name suggests, is the difficulty in breathing. So, in the structure of DPCC, the critical design is absence of unsaturation in fatty acid chains. To the uninitiated, the nomenclature of phospholipases can sometimes appear to be confusing. the best way out is to start with a recap of the early enzymology of these enzymes. Phospholipases are hydrolases. While lipases digest fats and oils, phospholipases digest some phospholipids. Liver and egg yolk are known to contain lecithins. Around mid-1950s, it was found that lecithin is digested in the gastrointestinal tract by an enzyme which is also present in the pancreatic juice. This enzyme was called lecithinase. It was soon realized that since lecithinase is hydrolyzing acyl ester bonds on the glycerol, there may be similar enzymes which will also hydrolyze phosphatidyl serine or phosphatidyl aminoethanol. Hence, it was proposed that instead of lecithinase, all such enzymes are called phospholipases. Another enzyme which removes fatty acid from the middle C atom of the glycerol skeleton in the lysolecithin was called lysolecithinase. Lecithinase and lysolecithinase were called lecithinase A and lysolecithinase B. Eventually, these were called phospholipase A and phospholipase B respectively. It was known that lecithinase occurs not only in animals, it also occurs in plant tissues, microorganisms, snake venoms, bee venom and scorpion venom. The physiological action of the venoms was for a long time thought to be due to lecithinase or a phospholipase A alone. The actual picture is quite complex to be discussed here. These venoms also contain toxins. Some X-ray diffraction work on the phospholipases, and these peptides are also slowly bringing clarity to the picture. Lysolecithinase B was also known to occur in animals, plants and microorganisms. The substrate for this enzyme are various lysolecithins, which are in fact prepared by using phospholipase A action on various lecithins. Lysolecithinase of the mold Penicillium notatum was used to obtain L-alpha glyceryl phosphoryl choline by Uzil and Hanahan in 1956. The older literature mentions lecithinase B. As protein purification techniques became more powerful, this preparation was actually found to contain both phospholipase A and lysolecithinase. Even in 1940s, another phospholipase was known to occur in mammalian brains, snake venoms and an anaerobic bacteria called Clostridium welchii. Clostridium welchii was known to microbiologists as gas gangrene bacillus. This phospholipase hydrolyzed phosphatidyl choline to phosphoryl cholines and a diglyceride. It was given various names: Clostridium welchii lecithinase, Clostridium welchii alpha toxin, Glycerophosphatases, lecithinase D. It was eventually called phospholipase C. During the same period, a phospholipase was found in higher plants like cabbage, carrot and cottonseed, which had different specificity towards lecithin and other similar phospholipids. This was found to produce alpha phosphatidic acid and the base linked by the phosphodiester bond. The enzyme at one time was called phosphatidase C, but was later on termed as phospholipase D. Its early enzymology was carried out with both peanut and savoy cabbage. Phospholipase D turned out to be more widespread than in just plants. Let us look at the current nomenclature of phospholipases. The bonds which are hydrolyzed by various phospholipases are as shown in the figure. Phospholipases in general have different roles in biology, which is in vivo, and in biotechnology as in vitro. In living animals, phospholipases are present in the intestinal juice and take part in the digestion of dietary phospholipids. They have a similar role of digestive hydrolytic enzymes in bacterial secretions and venoms. Phospholipases also are part of enzyme cascades and signal transduction. Phospholipases A1 and A2 remove fatty acids by esterase action. Hence, these are unable to act on ether bonds present in ether phospholipids like plasmalogens. The product is a lysophospholipid. Some phospholipases are very specific. For example, one acting only on phosphatidyl choline. Others are less specific. The crystal structure of phospholipase A2 from a rattlesnake Crotains atrox contains the enzyme to be a dimer. The subunits are identical. The pancreatic phospholipase A2 was also found to have a similar structure. The pancreatic enzyme, however, functions as a monomer. Produced as a zymogen, trypsin removes a heptapeptide from the N-terminal. The enzyme has a great stability. It retains its activity even in the presence of 8 molar urea. It shows substantial activity in the presence of 2% sodium dodecyl sulfate. A protein lipocortin has been found to be an inhibitor of phospholipase A2. This is a protein of molecular weight around 37 kilodalton and it is induced by glucocorticoids. The inhibition seems to be via its interaction with the phospholipid substrate. Phospholipase C hydrolyzes PIP2 to generate PIP3 and diacyl glycerol. The structures of the PIP3 and PIP2 are as shown in the slide. Both molecules act as secondary messengers in signal transduction. This phosphoinositide pathway is discussed in more detail in the signal transduction chapter. Phospholipase D has been found in bacterial, plant, yeast and animal cells. Some phospholipase D are quite non-specific and will produce phosphatidic acid and the other alcohol. Even sphingomyelins, phosphatidyl glycerol and N-acyl phosphatidyl in the ethanolamine have been found to be substrates. Mammalian phospholipase D1 and phospholipase D2 are specific for phosphatidyl choline and have been cloned. Other mammalian phospholipases Ds are specific for other phospholipids and they have also been detected in several other tissues. Phospholipases have proved very useful in modifying natural phospholipids to tailor their properties. Such phospholipids find considerable applications in plasma, in pharma, food and cosmetic industries. Both drug delivery and gene transfer technologies have utilized phospholipids as vehicles. In general, phospholipases can catalyze both hydrolytic and reverse reactions. Both hydrolysis and transphosphatidyl of lysophosphatidylcholine choline with phospholipase D preparation from Savoy cabbage was investigated. For reverse reaction, 2 molar glycerol was used, which helped in suppression of the hydrolysis and reverse reaction. However, unlike in hydrolytic reaction, detergents did not help the enzyme activity in the reverse direction. An example which further illustrates the synthetic utility of phospholipases was the synthesis of 1-lauryl glycerophosphate and 1-lauryl dihydroxyacetone phosphate by combination of phospholipase C and phospholipase D. A two-phase system of diethyl ether and water facilitated the recovery of water-insoluble products and hence was used as the reaction medium. In this module, we learnt about the structure of glycerophospholipids and their role, ether phospholipids including plasmalogens, lecithin as a byproduct in oleochemical industry, and we also learnt about phospholipases and their biochemical importance. Two important points are worth noting. First, phospholipids are just not constituents of the membrane. When released and hydrolyzed, they produce molecules which are involved in regulation of metabolism and signal transduction. Second is the importance of phospholipids and phospholipases in industry. Lecithin is, for example, is an established industrial grade emulsifier acceptable in food and pharma and cosmetic industries. Phospholipases similarly are useful as reagents to engineer new lipids.